Okay, so here we'll focus a bit more on the specific technologies or the specific approaches we want to take to accomplish those goals. And I think the general and more broadly specified goal is we want to test many variants with faster, cheaper, higher resolution quantitative phenotypes. And there's a particular directions we need to focus on in order to get there, right? One of them is we want methods to collect not just static snapshots of physiology and organismal measurements, but also temporal measurements. And I think there's some ideas out there on wearable devices that we can incorporate into our measurements. We want enhanced tools for perturbation assays. I think there's a lot of discussion about this. It's obviously clear that perturbation assays will be one way we'll approach this. And for that, we want long DNA synthesis, cost effectively. And there are enzymatic approaches we can consider. Delivery methods, I think getting these constructs into cells, often cells die when you try to bring this into cells. It's hard to get into tissues more generally, where we want to study these variants. And improving our delivery methods with microfluidics or other technologies could be a really big boon to this effort. We want large improvements in throughput, right? To test combinatorics and just large-scale variant dissection, we want about 100 to 1,000-fold improvement in throughput. And one suggestion was to use pooled measurements to maybe put multiple perturbations per cell, and then use computational tools to derive the functions of each individual perturbation you might include, and then follow up with more targeted measurement. New methods for telomere-to-telomere genome sequencing and functional genomic assays for repetitive regions. I think there's a roadmap to do long read sequencing, but perhaps there's going to be gaps even with these very long reads where we want to use imaging in order to get there, as well as to layer on functional data. I think imaging would be a good approach. And I think it's a mistake to think that we can foresee the technologies that we want to use even within the next 5 years. Technology development occurs very rapidly, as we all know. And I think we need to think more generally about how to enable technology development, how to bring that into our community as it gets developed, and keep ourselves open to things that might be occurring in other fields we might want to incorporate into these efforts. And then now going to the data and sample resources. First and foremost, I think it starts with the genotyping data, right? We want to be able to integrate all our genotyping data in one coherent place, to be able to use it in different contexts. And I think one emerging topic is this understanding that a lot of data will be generated outside of biological research, right? Stuff that happens in industry for other purposes. And I think learning to integrate across those different data sets, as well as learn to even how to collaborate with those efforts, I think will be something we'll need to learn as a community. We want accessible and queryable biobanks, tissues with electronic health records and consented. And I think it's really important to now establish these, not just for DNA and RNA measurements, but for things we might want to do in the future. So as new technologies come on board, we can go back to our original measurements and start layering on things like spatial information if that's where things are going. Resources for engineered models, right? It's pretty clear from these discussions, particularly for the perturbation-based assays, that we'll need cell systems, either from cell lines to organoids to organisms, and having just a coherent place where we can start to access that, perhaps cells and biobanks ready to go, or just methods that are available online will be important. Benchmark data sets to compare methods, right? Experimental methods, a place where we'll want to kind of do all these different CRISPR-based perturbations, make sure that they're working in the ways we expect. Computational tools to kind of optimize across these different layers, and as well as kind of a coherent data set to pose challenge problems where we ask people to integrate across these different layers and try to build more kind of realistic models, right? And points were raised, we'll want to recruit and incentivize members of the community to kind of organize these efforts, either through jamborees or bake-offs. And of course, we're going to want to do more genome sequencing, right? I think from these discussions, we're going to need more to do imputation, either across hard-to-sequence regions, things that are currently missing, or diverse populations, including ethnic backgrounds and gender. I think it's quite important that we fill in those gaps before we move forward.
Thank you.